Okay, good afternoon, everybody. Thank you very much for the introduction. So now let's switch from image processing to zoology. So today I will talk about chemical inducers of metamorphosis in jellyfish. And so that's jellyfish and cnidaria are my model objects. And so I will give you first a small introduction about them, and then about a little bit about our research about their life cycles. So in general, why is it interesting to study cnidaria in general? So there are several reasons for that. So first of all, that's this phylogenetic position of cnidaria. So it's a sister group to all bilaterian animals. So these are very ancient animals. And so they are evolutionarily old. So at the same time, they are morphologically relatively simple. And at the same time, they are genetically complex. So the number of genes in human and in jellyfish, they are more or less the same. So this animal group invented nerve cells. So they also have multipotent stem cells. They can regenerate very well. Some species are immortal. So you can study many, many things by using these animals. And there are several model systems. And so I'm using mostly jellyfish, moon jelly already, and also to some extent little bit hydra. So this is actually the typical life cycle of a jellyfish. So, and this is a very interesting animal group because actually this group also invented complex life cycles. So, and then what is interesting? So why this animal is especially fascinating? Because several, as you can see, several morphologically and physiologically different life stages are generated by one genome. So we have at the same time in one life cycle jellyfishes, which are motile, they reproduce sexually, they live not very long. So they have complex sensory systems, so like eyes, for example. And then we have another generation, so polyps, which are sessile, they reproduce asexually, actually can live forever, can regenerate very well, have very simple nervous system. And so, and all that in one life cycle. So one of the main questions is, how are these transitions from planula larvae to polyp to jellyfish organized? So how does it work? What signals are necessary for this life cycle? And so this is one of the topics which we study. And as you know, so jellyfish is not the only animal with a complex life cycle and some metamorphosis events. So as you know, like this tadpole to jellyfish transition or caterpillar to a butterfly. And so it's actually in jellyfish, it's more or less the same. There are different body forms which change from one to another, but this is actually the most ancient animal group which has this type of transition. And actually there are some cascades, and we already know which molecules and signaling cascades are necessary for this transition in some groups of animals. Usually the initial signal comes from nervous system, and this is the situation in frog. And then, so there is a thyroid hormone, and then it binds thyroid hormone receptor with a coreceptor, and the main signaling molecule which facilitates this transition is a thyroid hormone. So in insects, the principle is the same. Again, the first signal comes from a brain, then ecdysone is produced, then it binds ecdysone receptor. In this case, ecdysone is the main molecule, which is important to this transition. And in jellyfish, actually, nothing was known. But so now I just introduce you how it actually happens, this metamorphosis. So, and I will talk today about this transition from polyp to a jellyfish. So usually in the laboratory, we can keep polyps just in the glass jars at 20 degrees Celsius. And then we can induce this transition by temperature shift. So it's very easy to do. You take a dish with polyps, there can be hundreds of them or thousands, and you just simply put them in a refrigerator. And then after some time, very interesting thing happens. About after 3 weeks, they start this transition. And it looks like a wave-like process. So this is a polyp, and then it makes kind of a constriction on the top. And kind of a segmentation process starts, which goes from up to down. And then this small segment forms small jellyfish. So it's actually a very interesting process. And it seems that it's somehow regulated, and we wanted to know how it works. So what are the molecules? And then we did very simple and maybe stupid experiment. We thought, okay, so this is a segmented animal, and this segmentation goes from up to down. So what happens if we make transplantation experiment? We take polyp, and then we take this late strobilar stage and transplant it here. Is the signal to metamorphose contained in these segments? And so the procedure is not so easy. So you have a fishing line, then you put these pieces together, and then actually they heal after some time, maybe 3 hours. And then you remove fishing line, and these pieces hold together. And it was a very big surprise to us that after about 72 hours, so the metamorphosis starts. So instead of 3 weeks in the refrigerator, when you transplant, so you can induce this transition. But this, this experiment is very tedious, so it's difficult to transplant, and we were a little bit lazy. And so we thought that, okay, and what happens if you simply, you cut some pieces and you feed it to the polyp? So they are not really cannibals. So it's actually, this piece is not digested, just stays in the gastric cavity. But this functions actually very well, and you can induce transition, this metamorphosis, even after 48 hours. So it goes very quick. So that means that there is some substance inside which induces metamorphosis. So there is this also some molecule which functions as a metamorphosis inducer. And so, but surely if you do some control experiments, like you take a piece of a jellyfish or a piece of a polyp, so nothing happens. So, and this experiment showed us that, first of all, polyp to jellyfish transition is induced by temperature shift. That was known before. And then that the most important is that the strobilar segments, they contain endogenous soluble inducer, which is actually absent in the polyp and environment. And then it was just a technical thing to find potential molecules. So we did some microarray experiments and looked for the genes that are upregulated at the cold temperature, actually in the refrigerator. And also, this inducer must be expressed exclusively in the strobilar stage. So this is in situ hybridization. We got a number of candidate genes, and you can see that some of them are not expressed in the polyp, not expressed in the young jellyfish, but expressed exclusively in the polyp stage. And then we tested, looked what these genes encode for, and then actually it was a little bit difficult situation because they all express some secreted proteins, so it's a signal peptide, but then there were no any special features. So all these proteins didn't have any real blast hits, so there are no analogous sequences in other animals. So it was something kind of jellyfish specific. And that was a little bit so difficult to analyze further, but we didn't give up. And we looked also at the expression dynamics of these genes. And one of them, actually, immediately when you put animals in the refrigerator, it is upregulated. So this gene is switched on. And then the amount of transcript, actually, amount of this transcript, amount of the product is is correlated with the duration of time which animals spend in a cold temperature. So this gene kind of records the duration of a cold period. And then we also try to fool the animals. So we take animals from 20 degrees, put in the refrigerator, and then after 1 day, we remove them back. So, and if you do this like for 6 days, after you remove them back after 6 days, so they do not metamorphose. But if you keep it for the longer time, so you start the procedure, so they cannot return back to the polyp stage anymore. And so what is the meaning of this mode of function? So what this gene is doing? So it's kind of, if you put animals in a refrigerator, so the amount of transcript increases. When it reaches some sort of threshold, it works like a switch. And the system also records the time of a cold period. Why is it important? It's very important because of the reason that the animal must metamorphose at the proper time of the year. So they must do it in spring and not at some point in winter or autumn or in summer. And so the system actually is very similar in principle, in its logic, to the vernalization in plants. So as you know, now sakura is blossoming and it needs actually cold period in order to blossom. And this is exactly the same situation. Polyp also needs cold period in order to metamorphose. So polyps, actually, by using this gene, they measure the duration of cold period for proper lifetime control. And then, so how this protein looks like. So it has a signal peptide. So it has a lot of repeats, a lot of aromatic amino acids. And then, so we try to, it looks like a peptide. So most probably just a small part of it works. And then we did some experiments, and actually finally we found peptide which induces metamorphosis. But that's not the end of the story. When we looked back at this peptide, we realized that, well, this combination of aromatic amino acids, they look similar to the molecule which was actually shown a little bit before to induce metamorphosis. It was just a screening of chemicals by Japanese colleagues, and they found that actually anti-inflammatory drug in the vitacil induces metamorphosis already. This is really strange because, so this is a blocker of prostaglandin synthesis. And if you use the similar blockers like ibuprofen or something else, it doesn't function. And so when we knew what is the molecule, we realized that this is just kind of actually not a coincidence. This is actually pharmacophore of the natural inducer, which is in reality is a peptide. And then we started just to look at other similar components, also with indole, which are all indole derivatives. And then, well, the majority of them didn't work anyhow. So some of them were toxic, and there were 5 components which were active, so they could induce metamorphosis. And also they actually had a different power to induce metamorphosis. So, and this one was the best one, so it induces metamorphosis within 48 hours. And also this is a very strong molecule, so it works in nanomolar range, as you can see here. And it's actually, you need just 160 kilograms to reduce metamorphosis, in 1 cubic kilometer of seawater. So, and kind of as a result, so the findings is that, so the transition is controlled by small secreted protein, so that the change in the water temperature plays the major role. And also, like a side effect is that now we can control the life cycle of a jellyfish. It's very important for laboratory conditions because previously we needed to wait 3 weeks until polyp to jellyfish. Now we can induce in 48 hours. And also as a side product, we actually found a very strong pesticide. So if one needs to eradicate jellyfish in a bay, so you can add it and they all will metamorphose into jellyfish. And if you do it, for example, in autumn, so all this jellyfish population will die. So you can actually exterminate jellyfish if you want. But we don't know the side effects. So nobody tried that before because maybe one will exterminate something else. So I don't know. So, but surely there are still a number of things which are not known and what I would like to study in the future. So now by these experiments, we little bit extended our knowledge about the metamorphosis regulation in the animal kingdom. So we found the inducer, so it's actually epithelia, small epithelia peptide. Also, retinoic X receptor functions as most probably as a co-receptor for the receptor which binds inducer. So this is kind of a common thing in the animal kingdom. So in frog, retinoic X receptor is a co-receptor for thyroid hormone receptor, and here it's ultraspiracles, which is a co-receptor for exosome receptor. But at the moment, we still do not know what is the receptor for our original peptide, or probably this receptor should also bind 5-methoxymethylandole. So we don't know what is the receptor. And so that is one of the things which I actually, it's good to have good connections and contacts maybe and discussion with chemists, because we can try to pull maybe the receptor by using this as a bait, but one needs to synthesize molecule, so inducer with some spacer and maybe some biotin or something else, or magnetic beads. in order to pull this receptor out of the protein, maybe protein solution or lysine of Aurelia proteins. And so that's our kind of plan, and would be happy to have some feedback or potential collaborators on this project. And also, I just want to say thanks to the persons who were involved in the project. So I recently joined Academia Sinica, so in autumn. So before I worked for some time in Germany for quite a long period of time, and also nearby on Okinawa in the Okinawa Institute of Science and Technology. And so thank you very much for your attention.